Alright, let's talk about green fluorescent protein, GFP. It's originally cloned, or it's originally found in a jellyfish that has fluorescence. And people won the Nobel Prize a few years ago for using GFP for research and for scientific implications. So they took the gene from the jellyfish, put it in a plasmid, and cloned it. The vector can be used in bacteria, but it can also be used in all different other animals. As you can see in these pictures, there are many different ways of using this marker, this indicator of gene expression in different animals, in bacteria, and so many other applications. So GFP has been an absolute amazing tool to learn about expression patterns and so on. But let's talk about the vector and about the gene. And how do we as bioinformatician learn how to use it and how to exchange and how to modify it? Alright, so you can see the vector here in the middle. It's called pGlow, and it has the GFP gene cloned into it. But when you look at it on a molecular level, as we have done with our other genes before, you can see that there's just a couple of different genes. It's got the origin of replication. It's got an ARA gene, is a promoter region, and it's got the beta-lactamase gene for ampicillin resistance, so you can grow it up on plates that have antibiotics in it. But if I give you the task now to say cut out the GFP gene and clone in the RFP gene, the red fluorescent protein gene, how are you going to do that? So I want to have my bacteria not glowing green, but I want to have them glow red. So I'm going to give you the sequence of the plasmid. And again, just like what we've seen in all the other experiments before and examples, it's just a sequence of unidentifiable patterns. So you need another tool to use that sequence, to use that long sequence with all the different genes and all the different elements in it, and figure out where is your GFP, your green fluorescent protein, how can you cut it out, and how can you clone in another gene. Alright, let's do some more bioinformatics. I'm going to copy the sequence of the pGlow plasmid, and it's pretty long sequence. And I will paste it into a program called Open Reading Frame Finder, ORF Finder. And again, it's one of these fantastic tools where you just paste it in and say submit. You don't need to change any modifications, and it gives you a map of potential open reading frames. So you can see that there's these longer sequences of red, and these are the most likely areas that encode for genes. And you've just seen before there are 3 different genes actually in that plasmid. The smaller reading frames are just start and stop codons, but they're interrupted by stop codons. They're too short to usually code for genes. So you click on the gene, and this program is very cool. You just say blast or smart blast. You don't have to copy anything. It just gives you already the information for the protein. Again, this is not the nucleotide sequence, this is a protein sequence. And you say smart blast. It already translates it for you, and we'll figure out what this first gene is coding for. So this first gene is the ARA-C gene. And let's give some details. And this is here we go. Here we go. This is a phylogeny, which is not really important right now, but what it is, it's a regulator gene, and it allows arabinose, which is in the medium, to bind and then allow transcription for the GFP protein. Hey, you can again find information about the paper, the comments, what it does, and so on. So this is your protein sequence. But that's not what we're looking for. We want to find out where our GFP gene is. So this one, the first one here, was our arabinose inducer protein. Let's check ORF3. And let's do smart blast, protein blast, and this one seems to be our GFP protein. All right, so our ORF3 seems to be our GFP protein, and it's a fusion protein on a vector pGFP. So this is our gene of interest. Let's quickly check out our next gene, and you probably should know what it should be, because there was the arabinose inducer protein, the GFP, and the other one was our resistance gene, the ampicillin resistance gene. Let's see if that's right. All right, beta lactamase. So this is our resistance gene that breaks down beta lactamase antibiotics. So this is the right gene. So we found our 3 genes in our ORF Finder. Again, look at the largest pieces, just click on them and do smart blast, and you will find out what the genes are. All right, so this is the main task of this activity. You can continue obviously and then find restriction sites, cut out the gene, clone in another gene, but that's material for another video. All right, so this is ORF Finder. Put in your sequence, run the ORF Finder, determine what the different genes are, and you have all sorts of information about the genes. You know where they start, where they stop, how long they are, what reading frames they are. And just a quick refresh on reading frames. There are 6 reading frames, and that means that's because we don't know exactly where the gene starts. So if it's an AGT, it could start at A, it could start at G, or it could start at T. And also genes can be read from both sides, so it can be read from 3 to 5 or from 5 to 3. So our 6 reading frames, our program considers all these different possibilities, and that's the information we have. So again, really cool program, really simple to use. Again, imagine, you have this information here, and you have to figure out where does your gene start and where does your gene end. No way you could do that by yourself without the help of one of these amazing bioinformatics tools. All right, I hope you enjoyed and learned something from this video, and we will see you in the next round of Bioinformatics 2.0.